Next, let us bow our heads for a moment of prayer, which will be led by Dr. Ahmad Siddiq.
Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Ladies and gentlemen, before we begin, I'd like to invite you to join me in a moment of reflection and prayer. I read this prayer in Islamic way, seeking peace, guidance, and blessing for all. Please take a comfortable position, open your heart. If you wish, raise your hand in prayer. Attention, prayer. Allahumma inni wajjahtu wajhiya lilladzi fataras samawati wal ard hanifam muslimau wa ma ana minal musyrikin. Inna solati wa nusuki wa mahyaya wa mamati lillahi rabbil alamin. La syarika lahu wa bidzalika umirtu wa ana minal muslimin. Allahumma anta malik la ilaha illa anta rabbi wa ana abduk. Dalamtu nafsi waqtarabtu bidanbi faghfirli dunubi jami'an. Innahu la yaghfiru dunuba illa anta. Wahdini li ahsanil akhlaq la yahdiya li ahsaniha illa anta. Wasrif ani sayyi'aha la yasrifu anni sayyi'aha illa anta. Allahumma salli ala Muhammad wa ala ali Muhammad kama sallaita ala Ibrahim wa ala ali Ibrahim innaka Hamidum Majid. Allahumma barik ala Muhammad wa ala ali Muhammad kama barakta ala Ibrahim wa ala ali Ibrahim innaka Hamidum Majid. Oh Allah, I turn my heart and face to you, the one who created the heaven and the earth in truth, in submission, and I am not the polytheist. Indeed, my prayer, my sacrifice, my life, and my death are all for you, the Lord of the world. You have no partner. This is what I am commanded to, and I am those who submit to you. O Allah, you are the King. There is no God but you. You are my Lord, and I am your servant. I have wronged myself, and I confess my sin. Forgive me all my fault, for no one forgives sin except you. Guide me to the best of morals, for none guides us to best but you. Turn me away from evil and indecent conduct, for none turn it away but you. O Allah, send your blessing upon Muhammad and upon the family of Muhammad, and you send blessing upon Abraham and his family. O Allah, bless Muhammad and his family as you bless Abraham and his family. Indeed, you are praiseworthy and glorious. Amin. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Ahmad. Ladies and gentlemen, to begin our agenda, we will hear a welcoming address from the head of Research Center for Preclinical and Clinical Medicine, BRIN. Please welcome Dr. Harimat Henderwan.
Assalamualaikum warahmatullahi wabarakatuh. The Honorable Chairman of Research Organization for Health, Professor Niluh Putu Indi Darmayanti, Associate Professor Takashi Matsumoto, Dr. Astri Dewayani, PhD, speakers, researchers within the Research Organization for Health, ladies and gentlemen, good morning, welcome, and thank you to each and every one of you for being here today. Praise to be Allah Subhanahu wa Ta'ala for His grace and guidance so that we can gather today to participate in this meeting. We are very happy today, the Research Center for Preclinical and Clinical Medicine, Research Organization for Health, are able to present a webinar entitled Research in Helicobacter pylori, Microbiome and Host Response toward Precision Diagnosis and Therapy. This webinar not only features highly competent speakers such as Professor Takashi Matsumoto from Oita University, Japan, Dr. Astri Dewayani, PhD from Erlangga University, Surabaya, but also presents related research conducted particularly by researchers from the Research Center for Preclinical and Clinical Medicine, Research Organization for Health, National Research and Innovation Agency, BRIN. Ladies and gentlemen, we know microbiome as a characteristic of a microbial community occupying a reasonable, well-defined habitat which has distinct physiochemical properties. The microbiome not only refers to the microorganism involved, but also encompasses their theater of activity, which result in the formation of specific ecological niches. Since the 17th century, microbiome research has developed rapidly, marked by the development of new techniques and equipment that enhanced enthusiasm for microbiological research and caused paradigm shifts in understanding health and disease. Microbiome research is very important because the human microbiome plays a role in various aspects of health and well-being. The microbiome assists in digestion and immunity and also mental health. Additionally, the microbiome also plays a role in food production and environmental quality. By understanding the microbiome, we can develop biotechnological innovation that can improve human and environmental health. Through this webinar, we hope to explore more deeply and share understanding about the microbiome and its influence on health. Lastly, I would like to apologize for the various shortcomings in organizing this meeting. Thank you to all parties who have supported the organization of this event, and thank you to the speakers and the organizers. Thank also for Professor Niluh Putu Indi Darmayanti for fully supporting this webinar. Please enjoy the webinar and hopefully achieve what is expected. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Harmat, for your welcoming address. Next, we are honored to have the Chairman of Research Organization for Health BRIN, Professor NLP Indi Darmayanti, to deliver her opening remarks and officially open today's webinar. Prof. Indi, time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Very good morning to Honorable Head of Research Center for Preclinical and Clinical Medicine, Dr. Harimat, distinguished guests, respected colleagues and participants. Welcome to our speakers, Associated Professor Takashi Matsumoto, PhD, Dr. Astri Dewayani, PhD, Dr. Kartika Fauzia, PhD, and Dr. Reza Yuridian. It is a great honor to welcome you all to this scientific forum on research in gastric microbiome and host response toward precision diagnosis and therapy. Ladies and gentlemen, as we know, the microbiome is defined as the collective community of the microorganism and their genetic materials that inhabit the human body, forming dynamic ecosystem. In recent years, studies on the gastric microbiome have key pillar of the precision health, offering insight into how microbial balance influences immunity, metabolism, and therapeutic response.
I'm sorry, Prof. Indi, we cannot hear you. Maybe there's a technical problem. Okay, sorry for the inconvenience. There's a technical problem. So we will continue. Before we proceed to the next agenda, We kindly invite all speakers, moderators, and participants to turn on your camera for a group photo session. Okay, our host will take a few screenshots to capture this special moment. Please stay smiling on camera for a few seconds. Ready? I will start count for first slide. Ready for the host to take the screenshot, okay? 1, 2, 3. Next slide. 1, 2, 3. Next slide. 1, 2, 3. Okay. Thank you, everyone. Now we will proceed to the main session. The first presentation session will be moderated by Dr. Zulfikar S. Ulhaq, PhD. Before that, I will read his CV first. Dr. Zulfikar Syambani Ulhaq, MBiomed PhD, is a senior researcher at the Research Center for Preclinical and Clinical Medicine, National Research and Innovation Agency BRIN. He earned his medical doctor and Master of Biomedical Sciences degrees from Brawijaya University and completed his doctoral degree at Kumamoto University, Japan in 2017. His expertise lies in clinical genetics, visual sciences, and neuroendocrinology. His research focuses on the genetic and molecular mechanisms underlying neurological and hereditary disorders. So without further ado, we kindly invite Dr. Zulfikar to lead the session.
Thank you very much. Good morning, everyone. My name is Zulfikar, and it's really a pleasure to serve you as your moderator for the first session of today's webinar. First of all, I would like to thank to all the participants for taking the time to be a part of this event. But before we begin, a few brief reminders. Please keep your microphone mute during the presentation. And we will have the Q&A session in the end of the first session, and you can type a question at any time in the chat box. So now let's begin with the first speakers. The first speaker is Dr. Kartika. She earned a PhD from Oita University with extensive experience in the GI tract as well as Helicobacter pylori. She also published various articles related with the GI tract as well as Helicobacter pylori, as you can see in the slide. And today she will deliver a presentation titled Research on Helicobacter pylori in GI tract disease. So please join me, Dr. Kartika. And you have 30 minutes for your talk. Please, the floor is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you for the introduction, Dr. Zulfikar, and also thank you for the coming of all participants. I'm Kartika Afrida Fauzia from Research Center for Preclinical and Clinical Medicine, National Research and Innovation Agency. At this opportunity, we will share about the current research of Helicobacter pylori in gastrointestinal disease from the pathogenesis to precision management. So Helicobacter pylori is a gram-negative spiral-shaped bacteria that infects the mucosa. Sorry. Okay. So oh yeah. Okay, so probably we will come Prof. Indi Darmayanti, who previously have trouble in the internet, so I'll return to MC.
Okay. Sorry, we will have the opening remarks again from Chairman of Research Organization for Health, Professor Indi Darmayanti. Are you there? Hello, Professor Indi. Sorry, because there's a technical problem, so Indi cannot join us. So we will continue for the presentation by Dr. Kartika.
Okay. Okay, is it my voice can be heard, or is it clear?
Yes, we can see,
Okay,
we can hear.
so we will continue our presentation. Is it our slide? Is it my slide can be seen? Okay, so Helicobacter pylori is a gram-negative spiral-shaped bacteria that infect the human stomach, or especially on the gastric mucosa, and it can infect chronically during a person's lifetime, and it can be a modifiable cause of gastric cancer. Actually, and this infection can induce the inflammatory response of the host cells, which is why it is a concern for the human health. And it is also a cause of a gastric cancer, which is by IARC WHO called as a carcinogenic bacteria. And furthermore, this Helicobacter pylori currently infect 43.9% of the global population. So the interesting fact about the Helicobacter pylori infection is that different areas have different prevalence of infection. For example, in the high infection is occurred in the China and India, also in mostly East Asia regions such as Japan and South Korea. However, infections are lower in countries such as Indonesia, which is less than 15%, but very high, more than 75% in India and also in certain region in Africa. Even though the prevalence of Helicobacter pylori infection tend to be lower since 1980 to 2022, we still have a quite high burden of the H. pylori infection and also the disease related to it. A study by Park et al. Prediction study population by Park et al. 2024 reported that probably globally 15.6 million population will have a gastric cancer case from the people birth within that are born between 2008 and 2017. So the person at risk highest are in China, and the predicted gastric cancer case are high in China and India, but in Indonesia, although it is quite not so high compared to those other countries, but it still have around expected number of gastric cancer cases are more than 100,000 cases among the 47 million people. So these are also a public health concern for the Indonesia population. And how and why the Helicobacter pylori are concerning? Because there is the study introduced by Korea at 2012. He introduced the Korea cascade of carcinogenesis on how the chronic infection of H. pylori can actually induce the development of adenocarcinoma. So the 85% of chronic infection of H. pylori can still cause chronic gastritis, which is also disturb the life quality of the subject. And sometimes it also can be asymptomatic, but 0.1% of them can develop mild lymphoma. And around 10 to 5% of the person infected with H. pylori can develop the antrum gastritis and corpus gastritis. So as we know that our gastric anatomy, they have a location called the antrum area, cardiac area, and fundus area, and the location of the infection can lead to different disease. For example, an antral gastritis can lead to hyperacidity and can lead to development of duodenal ulcer, while the corpus gastritis can develop into the gastric ulcer, intestinal metaplasia, and furthermore, it can lead to the adenocarcinoma of the gastric. How H. pylori can lead to the gastric ulcer, duodenal ulcer, and also adenocarcinoma, because H. pylori have or secretes the virulence factor. These are the protein that can help the H. pylori adapt in the acid environment and also can attack the host as the defense response of the H. pylori. So for example, the first one is the most important one is the urease protein. This help as we know that in the stomach, the acid level are around 4, which is the contribution from the acid chloric. So this in reality that for pH 4 can melt any surface, but in our stomach it is used to digest our food. And despite this very unfriendly environment, these H. pylori can survive thanks to the presence of the urease enzyme. And the next one, this H. pylori also have this CAC-A protein. This CAC-A protein can be injected to the gastric mucosa, and then it can lead to the inflammation of the host, and also can induce the activation of the carcinogenesis pathway. And also, H. pylori have lots of outer membrane protein. This outer membrane protein, such as BAP-A, BAP-B, SAP-A, ALP-A, and also OIP-A, they help the attachment of the gastric epithelium, so they can survive for long periods of time without without being swiped by the hosts. Also, it has the flagella, which also caused reaction of the host. So this is also help the bacteria to move around and infected and go to the more preferable area inside the stomach. So therefore, so that is why H. pylori is a concern. Concern can be concern for the human health. And also how we can diagnose the H. pylori, or how we know that H. pylori is infect us. There are two different methods that are used: the invasive and non-invasive way. The invasive way used the endoscopy and biopsy, then evaluate the histology and perform the rapid urease test on the biopsy. This is the gold standard that can prove the H. pylori are really exist on the mucosa. However, there are also other non-invasive for the screening, especially for the screening purpose. They are the urease breath test, also the stool antigen test, and they use the molecular identification for the susceptibility. Therefore, despite all these methods, there are still drawbacks and in many areas because the tests tend to be expensive. So this development of diagnostic tools for H. pylori are still also domain of research currently. And our previous study also used the serum pepsinogen as the gastric cancer and gastric biomarker for Southeast and South Asia population. So the study evaluate the serum of the 600 subjects, 1,200 subjects, and it showed that the subject with peptic ulcer disease have significantly higher serum pepsinogen 1 and serum pepsinogen 2 level. And it is showed that the combination of the serum pepsinogen examination and H. pylori status examination can predict the occurrence or the risk of the gastric cancer development. Therefore, it is a good approach to prevent for the screening purpose, and especially in the area that have a low facility for the endoscopy test. So the next one, if we have this positive H. pylori test, we will have to treat the subject. So this figure and this pathway I cited from the Indonesian consensus of the H. pylori treatment developed by the Gastroenterology Society of Indonesia. And it really, the first line therapy are clarithromycin. And these clarithromycins resistance are currently emerged in many areas of the world. So therefore, they recommend to use the susceptibility test first before the treatment. And if the susceptibility tests are not available, we need to consider the prevalence of clarithromycin resistance between these. So the first line treatment consists of clarithromycin, amoxicillin, and PPI or PPI proton pump inhibitor or PACB. And these three combinations are used as the first line. If the subject are resistant to one of them or the prevalence of resistance are high, the different antibiotic are recommended as the second line treatment. And currently rifabutin are considered as the therapy for the failure of the treatment. If first line and second line treatment are failed, rifabutin are considered. So this other concern of the research in H. pylori are about the antibiotic resistance. So this antibiotic resistance mostly caused by the resistance due to the 23S RNA mutation. So for how this resistance can appear inside the stomach, it's because it can be the H. pylori itself from the environment already have the resistance. They bring the resistance from the outside. The resistance can be developed from the ineffective usage of the antibiotics. So as we can see here, that there are also different prevalence of antibiotic resistance around the world, because they have different habits or regulation for the antibiotics. For example, for amoxicillin resistant, most country they still have a low prevalence of resistance. However, for the metronidazole, the prevalence of resistance are quite high, even more than 50% in the regions such as in China. The primary resistance for metronidazole are 35.3% in average, and clarithromycin also 32.6% in average of the world, and also 13.2% for levofloxacin and 4.8% for amoxicillin. So this is a field of research that are currently, especially the mechanism of the resistance of each antibiotics are different. For example, in macrolides, their mutation in 23S RNA and probably the new mutation can involved. And also, for example, in rifamycin, mainly the mutation in the targeted genes, for example, like GRACE A, RPOB, and also the 1650S and 16S RNA for tetracycline can induce the antibiotic resistance. But there are also other alternatives that are not due to the mutation of the target genes, such as there is a biofilm formation. There are the efflux pumps mutation, also the cocoid formation, and also other method. That's why a previous study in Indonesia reported in Indonesia we have the data of the H. pylori that are collected from the 5 major islands in Indonesia and also in east part of Indonesia, and we evaluate how the biofilm formed among those Indonesian strain, and we found that around 20% of the strains have the high biofilm former, and when we test the susceptibility to antibiotic, it showed that the presence of biofilm can increase the resistance, especially for amoxicillin and also clarithromycin quite high. And this is what is biofilm look like. This biofilm is kind of mucose that cover the bacteria. Therefore, it prevents the antibiotics to enter the cell, and thus it increase the resistance and reduce the susceptibility and make it difficult to eradicate. And other study also, we also utilize the molecular method to identify the mutation of the resistance through the whole genome sequencing. So our previous study report the study from Bangladesh. So the subject from the gastric biopsy, of the subject, we isolate the H. pylori strain, and then we checked the resistance and perform the whole genome sequencing and evaluate the GR-A, GR-B, 23S RNA, PBP1A gene, and find the SNP or single nucleotide polymorphism that are associated with antimicrobial resistance. We observe the the well-known mutation, but we've also found a new mutation that has not reported before. So this is why we can utilize more next generation sequencing for finding new mechanism of antibiotic resistance, and further can help the finding of the new target drug. And also there is also a interesting finding that H. pylori really can involve in the gastric microbiome. So we will have a further explanation from our next speaker. He is my teacher previously when I studied before in OITA, and he will explain more. But generally, the H. pylori really can affect the microbiome or the ecosystem of the organism that lived inside the stomach, and this is called the dysbiosis, and this dysbiosis can actually enhance the development of the gastric cancer. So we also previously have we studied around in Indonesian population. We see in here that the presence of the H. pylori can decrease the alpha diversity of the subject infected with the gastroesophageal reflux disease. Therefore, this can worsen the severity of the GERD, and it can also be an opportunity for us to understand that the gastric microbiome are actually play roles, so that we can try to make innovation to probably we can target the gastric microbiome as the new treatment strategy. So therefore, we can prevent the gastric cancer by eradicating the H. pylori. So if we can find the H. pylori as early as possible and treat it as early as possible, it can prevent the development of the gastric atrophy and also metaplasia, as also already recommended by the IARC, and also the Gastroenterology Society around the world. So for the future direction, we have the precision. We may consider the precision therapy, which are genotype, such as genotype guided regimens, host pathogen microbiome profiling, and also we should consider the new treatment strategy that are less that have less side effects. Also, we should consider to develop vaccine to prevent the infection of the H. pylori. I think that is all my presentation. Thank you very much for your attention. I acknowledge Professor Yamoka from Oita University, Professor Miftausur from Erlangga, also Dr. Harimat Hendarwan from BRIN. Thank you very much. And I return the screen to moderator. Thank you.
Thank you so much, Dr. Kartika, for your presentation. I think we have to move on to our next speakers. We are pleased to welcome Associate Professor Takahashi Matsumoto from Oita University. He has strong background in the genome analysis, next generation sequencing, and also microbiome study. And today he will present about the microbiome analysis foundation and application. Please welcome Dr. Takahashi Matsumoto. You have 30 minutes for your talk, so the screen is yours.
Can you hear me? And
Yes,
can you see my presentation? Good.
yes, very well.
Okay, selamat pagi. Nama saya Takashi Matsumoto from Oita University. So today I would like to talking about microbiome analysis, foundations, and applications. So as the old professor knows, microbiota is the collective term of the microorganisms that living in our body. So microbiome is a genomic approach to describe these old microorganisms in our body. Since the establishment of the bacteria culture by the Robert Koch, more than a century has passed, then still only 1% of microorganisms are culturable. Even if it can be cultured, the visually identifying them is extremely difficult. So improvement in the genome analysis technology has enabled accurate the understanding of information about even the unculturable bacteria. The Human Microbiome Project conducted by the US NIH, which made the foundation for the microbiome research, consists two phases. In the first phases, sampling methods and sequencing protocol were developed, while the second phase established cohort study related to the disease. And the analysis pipeline were established. Then additionally, currently data center being established recently. In this seminar, I would like to share our research experience and introduce some workflow and procedure of the microbiome research. So first, we need to collect the samples from our subject. So an enormous number of the bacteria exist in our body, and sample can be collected from various parts, and particularly in the bacteria residing in the gut are most of the biggest number. So whereas the bacteria inhabitant in the stomach which we study are likely the less abundant. Then furthermore, bacteria community inhabitant each area have specific trends. So, and the presence of those resident bacteria plays a critical role in maintaining the homeostasis in individuals. Some methods for collecting the sample from various sites have already been established. For example, on the skin, techniques such as using the seal patch and to collect the bacteria from epidermis are also used widely. For gut microbiome research is very common. So there are collection kits and the method for the storing the stool using some Bristol stool chart to record some stool condition. Sometimes it helps for further analysis. So you can just see your reference paper. So sometimes we usually use this collecting containers, then give the patient and we collect the stool. Then in case of the stool sample, it is recommended to immediately store directly at the -80 to prevent DNA damage. In our stomach, the gastric research, biopsies, we need the biopsies and need to be preserved. And then various reagents also have been developed. So those reagents can be also stored at 4 degrees and -20 degrees. So these reagents are also helpful for laboratory with the limited storage conditions. Also, we establish some in-house transportation mediums. It should be stored -80, but this medium is optimized for the H. pylori culture, also microbiome study. So maybe Kartika knows how to make this transport medium. She must know that this is the protocol from our laboratory. So the next we need to extract DNA. It is recommended to add the bees beating method to breaking fibers, especially when you use the stool samples. Additionally, bacteria have many cell wall components that making it challenging to extract very limited DNA in high purity. The cell wall components differ between gram-positive and negative bacteria, which might present in your samples, right? So in particular, the cell wall of the gram-positive bacteria is very robust, and sometimes in correct extraction method, that carries the risk of missing the presence of those microorganisms. So that's why to address this issue, the method using the enzyme cocktail for lysing the bacterial cell walls have been reported, especially we need the caution is required when using the samples containing some aqueous or such the soil samples. So some case in those bacteria is very strong. Usually they have very strong cell wall. And also we need to think about the timing of the sample collection. That's very important. For example, the previous antibiotics use can significantly impact the subsequent analysis. This figure shows the changing in the bacteria community richness when antibiotics were administered for 4 days. So you can see that after 4 days, that the richness is very decreasing dramatically, then keep decreasing at the day 8, then even the day 42, still cannot recover the completely. So our cases, our study, we set the condition that there has been no antibiotics usage for 4 weeks prior. So then we make some criteria. So maybe you may need to your own criteria. And also it has been found that the bacteria species obtained can be biased depending on the DNA extraction kit which I used. So this figure represents the analysis result using the 5 different DNA extraction kits. Then highlighting that the DNA, the certain species indicated in the uncolored box, something here and there, those was not extracted from each of the kit. So example, this is the blue colors, maybe this the DNA blood and tissue kit can extract most of the bacteria except those kind of species. So for the researchers, they must avoiding use the multiple different extraction method within a single study. So each kit has its advantage and disadvantage. So making it challenging to choose the best one. So for your study, just select one extraction method and then extraction kit. If you want to add the enzyme cocktail, just do enzyme cocktail. Then don't mix together DNA from different extraction method or different preserve storage the method. And many of you will likely focus on the human research subject, right? However, microbes are present everywhere in the environment. So when we should understand that our main enemy is the contamination. It is important to use some disposable chip, prepare aliquots, and maintain a clean lab environment in your daily research. That is very important. And next, let's talk about the library preparation. It can often feel very complicated. So, but the key is to read the manual carefully. So in my research, I extracted DNA from stomach biopsies. So most of the DNA is the human derived from the humans. So like experiment which other sample like the stool. So it's same as other experiment. We need to amplify bacterial DNA by PCR. But for our samples, so sometimes it's quite difficult to amplify our target DNA. So such case, it is necessary to adjust PCR cycles or other parameters. So this PCR targets the bacteria 16S ribosomal RNA gene. So it's this PCR step requiring the two-step amplification. First, amplification of the desired some variable region of the 16S RNA genes, then followed by addition of the index sequencing or adaptive sequencing for detection on the sequencing platform. The choice of the variable region depend on mostly your sequencing platform. So, and also many researchers select the PCR target as the V1 to V2 or 3, and V3 and V4. So in our research, we target the V3 and V4 regions. It's also known that some bacterial species may be difficult to classify depending on the target region. So you can see that's the figure. This is that's from same sample. The left side target for V1 and V2 regions. The right side is the V3 and V4 regions. V3 and V4 can detect the red colored bacterial species. So, and also some platform, the sequencer can be sequenced full length of the 16S, the ribosomal gene. So this is the most powerful and accurate result you will get. So this is the primer design for Illumina platform. So this is the, like, as you can see, at the end, 3' end of the primer. So there is the sequence target to the V3 and V4 region of the 16S with an overhanging the sequence outside that to bind to the index sequence and some other overhanging sequence here. In the second PCR, amplifications occur using the primer that include the overhanging and index and adaptive sequencing. So this result in the this two-step of the PCR gives the amplicon which can suitable for the sequencing on the Illumina device and has some index and adapter. So the index sequencing enables the multiplex analysis, allowing for the sequencing up to 384 samples in a single run. So we can have the variety of the index sequencing is available. So furthermore, by customizing the sequencing indicated in the green, so the libraries compatible with Illumina devices can be created for your sequencing. For example, in microbiome analysis, that's why the 16S ribosomal RNA is target for the bacteria. Then it's also possible to conduct analysis target of the fungi as known as the microbiome, and also the virus referred to as the virons. So you can design the as you want. You can just change the sequencing. Then you can put the index sequencing, and also you can sequence on the Illumina platform. So the library preparation feel tricky and complex, but actually library prep is simple and easy. Intelligence is not needed. Just please try it. And there are various sequencing devices, so it is necessary to understand this characteristic. So usually most of the researchers use this Illumina platform. So if you target sequencing variable 3 and 4, the length should be more than 500. So it means the platform gives this 300 twice. So it means around 600 500 base pair, you can get it. Then the throughput means the data what you want to get. So you can just predict your target sequencing only 500 the base pair, and if you do the 96 multiplex sampling, then divide it by 15 gigabyte. Then this is maybe it's quite difficult to say good or bad, because this quality score is very nervous for those companies. This is just my feeling. So those two platforms can gave us a more long read. The so-called, this is the long read sequencer. That was short read sequencer. So that means those two platforms can get the full 16 years the microbiome analysis. We can identify by species level. This is by genera. So then the PacBio is the very high quality read we can get. So the quality itself, I'm very happy. Then if the 16 years full sequencing quality is, yeah, I'm okay. So then Oxford Nanopore technology is a little bit cheaper devices. It's cheaper than PacBio, but quality maybe. But just do the 16 years full genome. Maybe it's I can agree. But Illumina, the quality itself is good. That's just normal sequencing, very good. It's no problem. But for the V3, V4, sometime, because we need to get the 500 base pair. So it means each of the 300 base pair of the sequencing should be matched, but sometimes couldn't match. So we should, at that cases, we should do the sequence analysis only the half part of the sequencing. So that's why. RBTs, I didn't use that platform, but the quality itself, it looks okay. So that RBT is that kind of the same, the amplification method of the PacBio. Then get more get good number of the lead. But I think this same problem might be happens. So because this is the target size and then the sequencing size. Maybe it's sometimes quite difficult to gather that's the both end of the sequencing lead. Then actually that running cost and the machine itself, Oxford is the cheapest. But we should think about license and voluntary fee. That's very good idea. Then the PacBio, we need the data server, and also the RBT gives the 300 gigabyte. We need the storage server at least. Then the MySeq, Illumina, unfortunately, the MiniSeq, ISeq, the MySeq is available until the December 31st in 2029. So they will stop the service for those platform. Maybe you shouldn't buy the platform from now. Maybe you can buy other next sec or other devices from the Illumina. Okay, then about the data analysis, maybe I have very little bit need to quickly just data analysis. We need to perform the command base. There is the QIM chime to the platform and some data 16S database is available. So, but anyway, you can use Linux OS or Mac OS or dual boot Windows OS. So we cannot use some normal PC like, yeah, that's for Windows or some VirtualBox is necessary. And then for installing your software, maybe Docker is much easier. And then some Nextflow is now is available. The same like the Docker, so they try to make the environment. So to installing the Chime, just we first need the mini conda anyway. even the directly install the Miniconda or the via Docker or VirtualBox. Just first we need to install Miniconda, right? Then this is command. Just type on your computer, install Miniconda, install Time2. That's done. So there are many of the tutorials you can just see. So now the researcher have to sit in your chairs and maybe making more headache for the big data and some maybe near future we should learn about the deep learning AI programming. Yeah. This is the last question. I'm talking about those of the procedure. Which process is most important? That is, I can say it's the study design. This is most important. So this is just protocol, right? So everyone can understand, but study design came some teamworks and need more the thinking about it, because the human genome is only 1% different each other, but the microbiome genes is less than 50% of the share each other. So it's quite difficult to quite easy to find the difference of you and me. So sometimes this is the different microbiome. This is because you are diabetes and they are not diabetes, they are healthy. This is different. Of course, everyone is different. It's sometimes they have the biases. So please be careful. Then recently they have the many of the some indirect the relationship of the microbiome and immune therapies, and also the gut and brain microbiome axis, something like that. So we should think about our study design very carefully. And lastly, I want to explain that briefly introduce our recent research findings. In the stomach of the Helicobacter pylori patient, like this is normal when the H. pylori were infected, then that infection cause dysbiosis characterized by the dominance of Helicobacter pylori. This is kind of the this biosis is just changing dramatically the component, the alteration of the microbiome, those called the dysbiosis. Then even after eradication therapy, H. pylori, of course, this is treatment of the antibiotics. H. pylori was removed, but this dysbiosis is going to continue. So this lies expectation for the application of some probiotics in the treatment. For example, so anyway, after treatment, we never go back to the normal stomach. Okay, because H. pylori was the changing that environment completely. So since Helicobacter pylori infect during the childhood, right, we transmit the H. pylori mother or father to the child. Then it may be that which bacteria colonize the environment the first strongly influence the subsequent establishment of that environment. This is the same as suggested in some ecosystem. So first, some seed is coming the soil. Then after the 100 or 200 years, so the ecosystem is never changed, right? So this is the microbiome is saying which kind of the microbiome is coming first. So that's why that's some research that indicated that if the baby come by the C-section, the more risk for the asthma or something like that. So H. pylori might be the first the pathogen to the infect our stomach. So maybe this is kind of the same some hypothesis or strategy maybe can be used. Okay, maybe around 30 minutes, right? Okay, terima kasih. Thank you for your kind cooperation.
Thank you so much, Dr. Takashi, for your presentation. I think there is a few question. We have received few question on the chat as well as in the chat. I think the first I will read for Dr. Kartika. Like the first question is why in Indonesia has lower prevalence of gastric cancer? Is it related with the cuisine or a diet?
Oh, thank you for the question. Thank you for the question. So Indonesia has, so actually for this question, the development of gastric cancer involved the virulence, involved the host and also the agent factor. So from the agent factor, the study in Indonesia showed that the virulence factor in Indonesia are not as quote unquote dangerous as the agent or bacteria virulence from the East Asia region. That is the first factor. And also the second factor is the host response to the infection. So the host response can be from the genetic nature of the because it's really ethnicity and also the environment and also the inflammation reaction nature are really contribute really important. And also the third one is the environment factor. Yes, there are Indonesian cuisine that are less harmful compared to the other country. For example, like in some countries such as Mongolia, they drink, and they even drink with the salt. So they add salt in their drink. And in Indonesia, we don't have that kind of habit, and also less alcohol consumption could also contribute to the less infection that occurred in Indonesia. And for the second question, the potential peptide as the antibiotic, then that is also currently a field of the research that's still ongoing. So although there are still no actual ready to commercial drug, it is still on the level of the animal model, but it can be a promising novel drug in the future for the multi-drug resistant bacteria. Yes, so this can be an interesting field to have deeper thought. Thank you. Maybe it's clear.
Thank you. Maybe from there is one participant raised the hand. So maybe you can turn on your microphone and directly ask the question.
Right. Thank you for the opportunity. I have two questions for the first speaker and then one for the second one. So for Ms. Kartika, my question is, we know that infection is a part of immune depletion and cytokine molecule secreted during immune response. Do you think we can use cytokine profiles as a predictive biomarker for disease severity or kind of like therapeutic outcome of the bacteria that infected individuals? And the second one, in terms of cell physiology, what makes the bacteria can survive low pH in human gut? I think you mentioned about, but sorry if I skip it. And then the last question for the second speaker, I'm sorry, I forgot the name. Dr. Takashi, I'm sorry, I forgot your name. So yeah, the question is, if I apply your method beside instrument feature, like the technology, the advance of the instrument, can you elaborate more what key element or step in study designing that I have to pay attention for better outcome? I think that's all from me. Thank you.
Okay, maybe Dr. Kartika, you can answer like very shortly.
Yes, for the first question, the inflammatory, yes, we will hear more this about the host response, inflammatory response from in the second session. Dr. Astri will have lots of will explain about the immunology, but currently we already such a cytokines such as interleukin 8, TNF alpha are commonly measured the level of inflammation. And for the second question is apa yang kedua tadi?
Maybe can you repeat the
So
second question?
the second question is, in terms of cell physiology, what makes the bacteria can survive the low pH in human gut? What makes them strong?
Okay. So for the H. pylori, they have the outer membrane protein that can mimic the host. So this is one of them. So they have less reaction. They are not easy to swallowed by macrophage. That is the first. And the second one is the H. pylori are easy to mutate. They have the high recombination rate in compared to other bacteria. So they can change it, let's say, quote unquote, it's like change themselves to adapt to the hosts. So that's why they can survive in the long term, and even it even spread to other people and their descendant, like vertically and horizontally.
Thank you. Is there any comment from Fendi? If no, then maybe Dr. Takahashi could answer the
Yeah.
question.
Okay, thank you. Thank you for your questions. So first, I want to answer the Mega, Mr. Mega. So how big does the role of probiotics to prevent complete against H. pylori? So to prevent Helicobacter pylori infection, please wash your hand. That is most effective prevention, because this is just from your stool then to your child. So just wash your hand and don't give the food by hand, and well cooked. That's enough. No need to give the probiotics for the baby. But some study indicates the fibers and some fruits with vitamins are kind of the factor for the prevention. And yeah, maybe even in Japan, even after the treatment of the Helicobacter pylori, 20% of the patient might be cancer or some other diseases. So those cases, maybe we think about the probiotics. Then some dysbiosis occurred after treatment. So the remained bacteria still looks the virulent bacteria, and also we should maybe manage those the virulent bacteria away from your stomach. Okay, then the second other question, the contamination DNA extraction. Just select the one method. That's enough. So I just want to say that don't mix the two extraction kit in the experiment. For example, you have the case and control. The case extract the QIAGEN kit, then control extract the ProMega kit. That is completely wrong. But you can just select one, then keep using that one. Okay. Then Fendi, Dr. Fendi, what is the Fendi Puradana? What was your question? I couldn't catch that. Your question.
Okay. Hi. My question is, if I apply the method, because you explained more about the technical stuff in the lab. So if I apply the method beside instrument feature, like the advance of the instrument, can you elaborate more what key element of stuff in study designing that I like really have to pay attention on it for better outcome of my study in terms of working with this bacteria?
Oh, wow. It's quite difficult, this question. So, a difficult question. Maybe you should you should discuss the researcher from other field. Yeah. So, and then, for example, I'm very questionable from the concept of our laboratory. For example, some students want to compare the microbiome from cancer and healthy stomach. So there are some meaning, but if you do the endoscope, maybe you don't need to do the sequence, right? The medical doctor can see, oh, this is cancer, this is healthy. What does meaning? Yeah. Why we need to do the sequence? Why we need to do the microbiome analysis? So this is kind of just a method. So microbiome is just a method. So that's why we should think about that. If easy to distinguish the two groups, no need to do the microbiome study. So maybe this is just the message for you. Thank you.
Yeah, thank you.
Thank you so much. If there is no question, maybe I have one question for Dr. Takashi. Like you mentioned before that we can select only one kit, right, for the extraction method. And then is there any preference, like which extractions, for example, it's much better outcome or like than the others? Or maybe you have some advice, or maybe you have some, I don't know, maybe expertise on it.
I prefer use the QIAGENs. That's QIAGEN.
Okay, is there any like specific to say the material why for example like QIAGEN give like more better outcomes
Because
than
more
the
than 10 years, our laboratory used the QIAGEN kit for extracting the DNA.
Mhm.
So we shouldn't change. Otherwise, we cannot compare the data
I
from the
see.
older days. That's why. So maybe it depends on your laboratory conditions.
Mhm.
So we cannot say that which one is the best, but you should select. You should understand the background and lab. Yeah, just select. Keep using the same kit. It's not so big problem. It's okay.
Okay, thanks so much. I think maybe this is the end of the first Q&A session for the first session. And before we move to the next session, on the behalf of the organizing committee, It is my honor to present the certificate of the appreciation as a token for recognition as a speaker for today's webinar. So you can see this the certificate on the screen. And I think that concludes our first session today. Once again, thank you for all the speakers for your great presentation. And now I'd like to hand the session back to you for the next agenda. Thank you so much.
Thank you, Dr. Zulfikar, for leading the first session. Next presentation session will be led by Dr. Pramita Andarwati. Before that, I will read her CV first. Dr. Pramita Andarwati is a junior researcher at the Research Center for Preclinical and Clinical Medicine BRIN. She holds a medical doctor and a master degree in immunology from Erlangen University with expertise in healthcare and integrative medicine. Her research explores the health impacts of kratom consumption, rational use of antibiotics, and the integration of traditional and complementary therapies such as acupuncture in healthcare practice. So without further ado, to Dr. Pramita, time is yours.
Good morning, everyone, and welcome to the second session of today's webinar. My name is Pramita, and it is my pleasure to serve as your moderator for this session. Before we begin, I'd like to extend our sincere appreciation to all our speakers for joining us, and to everyone in the audience for taking the time to participate today. In this session, we are honored to have two distinguished experts, Dr. Astri Dewayani, PhD, and Dr. Reza Yuridian Purwoko, who will share their insights and research on host responses in gastrointestinal diseases and the gut-skin axis. We will have a Q&A session at the end of both presentations, so please feel free to submit your question at any time using the chat feature. Now, without further ado, it is my pleasure to introduce our first speaker, Dr. Astri Dewayani, PhD. Dr. Dewayani is a lecturer and researcher in the Department of Anatomy, Histology, and Pharmacology at Erlangen University. Her research focuses on cancer immunology, the microbiome, and animal modeling. She earned her medical degree from Erlangen University in Indonesia and completed her PhD at Oita University, Japan. Dr. Dewayani's extensive work on Helicobacter pylori and its role in disease mechanism has been published in several well-regarded scientific journals. Today, Dr. Astri will deliver a presentation titled Evaluating Host Response in Gastrointestinal Disorder. Please welcome Dr. Astri Dewayani. Dr. Astri, the screen is yours.
Okay, thank you for the introduction, and I'm very honored to be here to present in this webinar. So let me share my screen. Okay. So in this occasion, I will share a bit about what I've studied, especially with Dr. Kartika also in Oita about the host response in GI disorders. So first, we already know that we have two types of immune cells. So from the stem cells, it will divide it into two, myeloid progenitor cell and lymphoid progenitor cells. The myeloid will become the innate immune cells, which include dendritic cells, macrophage, and then PMNs, and so on. And then for the lymphoid progenitor cells, it will become adaptive immune cells, which includes T cells, B cells, NK cells, and so on. And then through the first infection, either it's parasites or bacterial. So the first one is innate immune cells will recognize, and then later it will become the APC or antigen presenting cells, and then it will present into the innate, the adaptive immune cells. So it becomes specifically mature by the receptor. And then for the APC, there are two, dendritic cells and then macrophage. And then they will recognize PAMs, which is called pathogen-associated molecular patterns from the pathogen or from the dead cell, which is DAMs, damage-associated molecular patterns. After recognize it, especially PAMs, usually it's coming from the outer bacterial cells, and then recognized by one of it is TLR receptors. And then TLR receptors, this complex, later will cascade and then trigger the NF-kappa B from the innate immune cells, disease, and then macrophage, and then later they will produce cytokines, and then those cytokines will be present into the T cells, and then what kind of cytokines will then decide the differentiation of T cells. And later we will see that what many types of TH subsets. Okay, so for Helicobacter, because it's a gram-negative, and then so usually it's through TLR4. This is the classical LPS receptor, TLR4. However, besides that, the non-classical, they can use TLR5 from the flagell, and then also TLR2. Okay, so the TLR, this is the classical way. It's the TLR4 will combine with MD2, and then later activate NF-kappa B, and then this, as being asked before, that what kind of biomarker. So the classical or must be increased in level. Usually are IL-6, IL-1 beta, TNF-alpha, and also IL-8. It can be from the innate immune cells, or also increase from the gastric epithelial cells. However, H. pylori has a different mechanism when it's LPS or antigen express Lewis antigen that mimics human Lewis molecules. That's why it can facilitate the addition, enable evasion, because it mimics the human Lewis molecules. And then, as mentioned previously by Dr. Kartika, that's why it can evade macrophage phagocytes, because through this mimicking human Lewis, it's to making a macrophage towards the M2 types, where the macrophage has two subtypes, M1 and then M2. So the M1 usually more into clearing and then pro-inflammatory, while the M2, it's called TAMS, or tumor-associated macrophage. So it's usually more into the anti-inflammatory, and then towards to the oncogenesis. That's why it can help the bacteria to evade the phagocytes. Okay, in these schemes, we can see that the Helicobacter pylori can attach, as mentioned before in the first session, and then they can directly express arginase that, and then also that Lewis molecules, mimicking, so that's our macrophage towards into the M2 type macrophage, which is later also give cytokines that for the PMN recruitment. With the PMN recruitment, it's also increase the inflammation process. While the dendritic cells, it's elongate, and then taking the antigen from the pylori, and then later will present as the APC when if it's produce more the IL-12, it can stimulate the naive T cells to become Th1. And Th1 itself mostly produce interferon gamma, and then it's also activate the macrophage into coming into the environment. While dendritic cells presents the IL-23, and then it's helped to differentiate the naive T cells into become Th17. Like why this is here, it's called the Th17 pathogenic. Later we will learn that actually the Th17 normally is stimulated by TGF-beta and then IL-6. However, if it's stimulated by IL-23, it can shift towards the different type of Th17. Okay, so the Th17 will produce IL-17 and then IL-21. While IL-17 usually it's good for the acute inflammations together with interferon gamma, because we need actually the inflammations to clear up the infection, bacterial infection. However, later, what we call chronic inflammation, it's actually a shifting balance of immune cells production. So if the production is too many to handle, and then it can be lowered, and then balance. So later it will becomes chronic inflammation. And so the IL-17 for the acute inflammation, it's good because it's also clear up the bacterial. And then the IL-21, it's autocrine to maintain the Th17 production. And then it's also can be stimulate the Th1 cells. While Treg or Th regulator, it's produce IL-10. Now, the IL-10 has anti-inflammatory effect. So that's the one balance the conditions. So the inflammation, it's not enormous too much, and then it can be balanced. And then besides that, the B cells also produce antibodies, the H. pylori antibodies to attacking the inflammations, the infections. So the first time, the acute inflammations, and then it needed. However, later when because H. pylori has the mechanism to evade the acute inflammations, and then mostly asymptomatic. That's why later it can be towards into the chronic inflammation because the infection still persistent, and then the TH balance, especially TH17, and then T regulation balance is shifting. Okay, so adaptive immune cells, the APC, as mentioned before, the APC will present the antigen and then the cytokine to the naive T cells, and then different cytokines will decide into different subtypes of T cells. So IL-12 usually towards the TH1, and then IL-6, and then IL-21 for TH17, the usual one. However, if it's stimulated by IL-23, usually it's become TH17 pathogenic, which mostly produce the IL-17F, not A. And then on the TGF beta towards the T regulator that produce IL-10. So each different type, subtype of T cells has also a regulator gene, different regulator gene. So TH1 is Tbet, TH17 is ROR gamma, and then Treg is FOXP3. And then this is not only the subtypes of T cells. However, for the gastric inflammation in in pylori infection, those that's mostly extensively studied. However, different type of TH, such as TH22, TH9, and then TH2 also there. So before, in the beginning of studied about the T cells, everyone's found that there are two types of T cells, which is TH1 and then TH2. However, later, the TH2 subsets, especially divided into different, such as TH9, TH17, it's actually not differentiate, but it's when they know that the gene is different and then the cytokine is different, and then they name it also different. And then for TH1, because it's produced interferon gamma and it's mainly known for also in the inflammation conditions. So firstly, they said that it's mainly depends on the TH1 response. And then because it's activate the M1 macrophage for the phagocytosis and then to kill the bacteria. However, as we already know that if the shifting balance, if the balance shifting and then the inflammation continues, and then it creates a mucosal damage, and then later will become a chronic inflammation. And then for TH17, it's known to be together with T regulations. So because the TH17 needs for the acute inflammations, because in the deficient, IL-17A and F showed lower inflammation in acute infection, while without the IL-17 receptor deficient mouse, and then it's significantly increased to chronic inflammation. So that's why we already know that IL-17 is needed, and then it's balanced, so it's not become overpowered by Treg, which produce IL-10. Okay, but the other studies saying that if the Tregs balance or it's in the chronic inflammations, and then Tregs is increasing with the IL-10 increasing, it's become towards anti-inflammatory. It damps the bacterial killing activity, and then later it becomes a chronic infection, and then the bacterial still there, the bacteria. And then from my previous study, it's towards the IL-17, IL-21, and IL-23 axis, because it's important because IL-21, as mentioned before, it's also for the autocrine, so it's help to maintain the Th17 differentiation. However, as pleiotropic cytokines, means it has dual site. The IL-21 also induce the MMP2 and MPP9 in the gastric epithelial, which related with the chronic inflammation. So it means that IL-21 is needed for the killing bacteria in the chronic infection for the Th17. However, it's also induced towards the chronic inflammation. While IL-23, as mentioned before, that it drives the Th17 to produce the pathogenic site. So that's why it towards the chronic infection. And then those similar in the GI disorder, the other GI disorder, because in IBD also the Th17 is increasing, and then because the balance towards the Th17, it is said that sometimes it might be that the people with IBD will have less infection of H. pylori because they have a higher IL-17. So in the first acute infection, to clear up the bacterial, it's easier. In the first session, we seen that different region has a different gastric cancer, and then we already know that H. pylori becomes the potential risk of gastric cancer, and then it might be also coming from the host response, because a different type of virulence, Eastern CAC-A or Western CAC-A, it will produce a different response. And then our body also, with the polymorphism of the cytokine produced genes, it can also associate with different prognosis of gastric cancer. And then the meta-analysis shows that polymorphism of IL-17 and then IL-23 is associated with gastric cancer, especially in Asian region. So the IL-23 receptor, polymorphism in it, linked with unfavorable prognosis of gastric cancer, while for the IL-17A, it's kind of still contradicting, because in the beginning we need the IL-17. However, if it has polymorphism and with a higher level, also identified as significant risk factor for cancer development. And then besides the two of it, the IL-1 beta polymorphism also plays role in the gastric cancer risk. And then because IL-1 beta, it's produced by the gastric epithelial cells, and then it's still contradictive that it might be linked with decreased risk or associated with higher risk. So because it depends on the region itself, so we don't have the data in Indonesia. How about the polymorphism? Is it also related with our lower gastric cancer prevalence incidence or not? And then besides the adaptive immune cells, they have the counterparts which called innate lymphoid cells. So it's actually has a similar progenitor. However, the innate lymphoid cells, or ILCs, it doesn't have the specific receptor to differentiate like T cells. So it can looks like a innate immune cells but it can express certain cytokine, specific cytokines like adaptive immune cells. And it's really specific tissue residence. So it means that different sites of GI tract has different ILCs, like in gastric. Our lab more likely doing it with anisakis, worm infections. And then it shows that in gastric, as either it's human or mouse, the gastric has more ILC2s, while in the intestine, more towards the ILC3. And then as we seen here, that the ILC2, ILC1 similar into TH1, and then ILC2 similar with TH2, which produce IL-4, IL-5, IL-9, and then for the ILC3, it's similar with IL-17. And then this why we have the innate immune cells when we have the adaptive immune cells. This ILCs is more robust in the infant age, when our adaptive immune cells, T cells, is not ready yet or immature, and then the ILCs that help our body to work like the adaptive. And then when we getting older, the ILCs become fewer, and then our adaptive immune cells will has a higher level. However, still the ILCs is still create in our adult age. So that's why some studies now investigating the ILCs number in the several cancer, and then the association with the oncogenesis. Okay, in the pylori infection, so the ILC will trigger the B cells to produce IgA, and then the ILC2 mostly activated by the IL-7 and then IL-33. And then ILC2 can also skewing the macrophage into the IM2 polarizations, similar to H. pylori, the beginning, with the Lewis. So the M2 type of macrophage can promote oncogenesis because it's more likely towards the anti-inflammatory. And then the question now is, targeting immune cells becomes a new therapy, while for the immunotherapy, they usually use the modified immunotherapy like CAR T cells, or even using inhibitor for certain cytokines. But is it really potential? We already know that the IL-17 and then interferon gamma are important for bacterial clearance, especially in the acute inflammations. And then the IL-17 subtype, IL-1A and IL-17A and IL-17F are extensively studied, and then it is said that IL-17F tends to play pathogenic site, and this IL-17F is activated by IL-23. So some studies have using IL-17 inhibitor in a different autoimmune condition, such as rheumatoid arthritis or psoriasis. However, it has adverse effect, which can increase the GI disorder. That's why when we choose to targeting what's lowering the IL-17F as a pathogenic site, which is IL-17, and then they choose the IL-23 receptor P19 subunit, because IL-23 receptor has two subunit. One is similar with IL-12, so IL-12P40, and then one is IL-23P19. So if targeting the whole IL-23 receptor, because it has the similar subunit with the IL-12, and it's indirectly affecting the number of Th1, because IL-12 is for the differentiation of Th1. So if it's only targeting the P19 subunit and then lowering the IL-23, it can controlling the pathogenic IL-17 response, and it's already been done in inflammatory bowel disease clinical phase, showing a promising potential. So we're not really clearing the whole IL-17 because it's still need in the GI, the whole GI, but it's only lowering the pathogenic side. However, for the gastric conditions, IL-23 inhibition has not been studied yet. So, however, theoretically, because it's the IL-23 levels is higher in the gastric cancer invasion, so probably it can also can be a potential adjuvant therapeutic strategy. And then for ILC2, the innate lymphoid cells, because it's just recently being studied, so still not many data about it, but theoretically it also has protumeric effect, so probably it can be targeted besides the well-known T cells immunotherapy. However, in adult, the ILC is really a small number. And then
sorry, Dr. Astri, you have 5 minutes left.
Okay,
Thank you.
so this is the last slide. So human body, it's really complicated. And then besides the direct cytokines production that has been mentioned before, don't forget that we still can learn about the epigenetic modulations, especially microRNA. So the infection of H. pylori, it can not modified, but it can induce certain gene, and then the gene can be translated into microRNA. Not messenger RNA is the one that being translated into protein, while microRNA, the one that stop or continue the translation, the copy of messenger RNA. And several, not several, so many, a lot of microRNA has been studied. So which one will towards the pro-inflammatory, or which one is towards the anti-inflammatory? So there's a lot more to discover for the immunogenic sites of the infection, and then also towards the oncogenetic. Okay, that's all from me, and then later, please, we can continue through a discussion section. So I will give back to the moderator.
Very much, Dr. Astri. Very comprehensive presentation. Now, let us move on to our second presentation. Our next speaker is Dr. Reza Yuridian Purwoko, dermatovenerologist specialist, a senior researcher at the Research Center for Preclinical and Clinical Medicine, National Research and Innovation Agency. Dr. Reza earned both his medical degree and Doctor of Biomedical Sciences from the University of Indonesia. His research focuses on regenerative medicine, and he has published numerous scientific papers and holds patents related to adipose-derived stem cells and also gene editing therapy. Today, he will deliver a presentation titled Gut Microbiome and Skin Health, the Gut-Skin Axis. Please welcome Dr. Reza. Dr. Reza, the screen is yours for 30 minutes. Thank you.
Thank you, Dr. Mita. Good afternoon, everyone. I will share my slide. So I'm a dermatologist. I'm a researcher with the Regenerative and Precision Medicine Research Group, Research Center for Preclinical and Clinical Medicine BRIN Indonesia. Thank you very much, Prof. Indi, Dr. Harimat, Dr. Ida, Dr. Tika, PhD, and the committee for inviting me. My clinical experience as a skin doctor of more than 15 years continually intersect with a simple set of patient questions. Dok, what should I stop eating to improve my acne or jerawat? Ya, Dok, ya. Dok, is my child's pruritic or itchiness, erythematous papules, allergy? What should he stop eating, Dok? Lots of questions around the gut and skin. Yeah. So, how does the gut health affect the skin appearance? The gut-skin axis describes the two-way communication between the intestinal microbiome and the skin. Trillions of gut microbes calibrate systemic immunity and epithelial barrier function through metabolites, such as short-chain fatty acid, bile acid derivatives, and tryptophan catabolites, and by training mucosal immune cells that circulate to peripheral tissues, including the skin. When the system is perturbed, gut dysbiosis, we call it, yeah, we see increased intestinal permeability, low-grade endotoxemia, and skewing toward T helper 2/17 inflammation. Clinically, that biology tracks with higher risk, greater severity of atopic dermatitis, psoriasis, acne, and rosacea. These links have motivated trials testing dietary patterns, abiotic to postbiotics, broader microbiome modeling strategies, and adjunct to standard dermatology care. The promise is targeted microbiome-informed therapy. The challenge is distinguishing correlation from causation and defining which patient will benefit. A 90 years old hypothesis helps frame today's science. In 1930, Stokes and Pillsbury proposed a gut-brain-skin connection. Psychological stress could alter gastric secretion and gut microflora, increase intestinal permeability, and drive systemic inflammation that manifests in the skin, acne included. They even suggest dietary measures as lactobacillus acidophilus and potential adjuncts. Clinically, early observation notes that 40% of acne patients show hypochlorhydria, consistent with altered hyper-GI ecology, while those ideas were ahead of their time and methodologically limited. Now, modern science validates key elements: stress HPA axis signaling, dysbiosis with barrier dysfunction, and endotoxemia, and downstream T helper 1 to T helper 17 neurophilic inflammation relevant to acne or other dermatosis. What has changed is we now investigate this pathway with microbiome sequencing, metabolomics, and control trials. The original concept was patients, to define which patient, which microbial target, and which intervention like diets or other modulators to standardize the dermatology care. How does the gut talk to the skin? Several converging pathways are involved. First, barrier integrity. A diverse eubiotic microbiota maintains a tight mucosal firewall that limits microbial and toxin translocation. Key commensals Lactobacillus, Bifidobacterium, and Akkermansia reinforce tight junction and mucus and promote regulatory immune tone. Second, metabolites, beneficial microbes generate short-chain fatty acid and other postbiotic that strengthen epithelial junction. Third, immune education and trafficking. Immune cells prime in the gut. Tregs, T helper substrate, innate lymphoid cells circulate systemically and influence cutaneous inflammatory set point. When this biosis occurs, loss of keystone commensals with expansion of pathobionts, permeability increases, low-grade endotoxemias, wing, emerge, and microbial products plus neuroendocrine mediators enter the bloodstream where they can amplify skin inflammation and barrier dysfunction. This framework explains why microbiome-directed strategy may serve as a adjunct to standard dermatology care. Two microbial ecosystems constantly talk to each other, the gut and the skin. The gut microbiome, trillions of organisms with over 3 million microbial genes, is dominated by Firmicutes, Bacteroidetes, Lactobacteria, Proteobacteria, and calibrates systemic immunity and metabolism. The skin microbiome covers our largest organ, for up to 2 meters square across oily, moist, and dry niche, performing physical, chemical, and immunologic barriers. Atopic dermatitis begins early and so can gut dysbiosis. Cohort studies show infants who later develop AD or atopic dermatitis have lower gut microbial diversity and delayed microbiome maturation versus healthy peers. This abiotic to dysbiotic shift linked to impaired function of the skin for a barrier and skewed to helper 2 T helper 17 immunity, supporting the rationale for microbiome targeted prevention or adjunct therapy in atopic dermatitis. Probiotics as evidence-based medicine, recent meta-analysis of RCT shows modest cohort reduction versus placebo, with largest effect in moderate severe atopic dermatitis. Signals are strongest for Lactobacillus salivarius, followed by Lactobacillus acidophilus plantarum, while fermentum shows little or no effect. So take-home probiotics can be as an adjunct, not a replacement, and selection should be strain and dose-specific, realistic expectation. Taken together, RCT indicate that the selected probiotic strain can serve as adjunct therapy in adult atopic dermatitis, the skin disease like itchiness and pruritus, likely via gut barrier reinforcement and immune modulation. Benefits appear independent of IgE or eosinophil changes. But severe scores improve. The LQI gains are inconsistent, and we still lack of clarity on optimal dose and duration. Bottom line, expect a modest strain-specific effect, strongest in moderate severe adult eczema alongside standard care. So the guideline is still working. We still need the RCT of the data of the clinical trial to make the standardized protocol for dermatologic condition. So this one is a VMT or fecal microbiota transplantation, a double-blind RCT in 2025 in moderate or severe adults showed that 3-weekly oral FTT dose produced larger ASC reduction and higher ASC 50 rates than placebo without serious adverse event. Immune profiling showed the reduction of 2 helper second and 2 helper 17 and other marker activity, and the gut microbiome shifted toward healthy donor profiles. What about psoriasis? Psoriasis patients show gut dysbiosis as well, reduced diversity with depletion of Akkermansia and some bacteroides, and frequently cluster with metabolic syndrome and sometimes intestinal bowel disease, supporting a systemic inflammatory axis. Most transfer studies suggest causality. Prostatic microbiota can worsen skin inflammation in recipient. Clinical meta-analysis in 2024 of 7 trials of RCT of 400 subjects found modest PASI improvement and CRP reduction with oral probiotic as add-ons. PASI 75 trended higher with no significant signal compared to the control groups. So it can be adjunctive as well, be put in the guidelines. How about acne? Acne, while driven locally by follicular keratinization, oily skin, C. acnes, systemic input, diet, stress, but also gut dysbiosis can amplify inflammation. High glycemic dairy patterns elevate insulin or IGF-1 and perturb the microbiome. Stress increase permeability and immune activation. Genetic epidemiology suggests causal link between specific gut taxa and acne risk. Early RCT shows symbiotic probiotic can reduce inflammatory relation and shift the gut toward the SCFA, producing species through effect of strain and study dependent. Long course of tetracycline disrupt gut flora, supporting consideration of probiotic therapy. How about rosacea, a severe form of acne? Rosacea frequently coexists with GI issues, especially SIBO. Cohort and interventional data show a meaningful subset. Test SIBO positive, and also the H. pylori link is debated. Some studies suggest association and improvement after eradication, others do not. Any benefit may relate to reduced TNF-alpha or interleukin-8 signaling. Clinical pathway, keep standard rosacea therapy as the backbone. Screen for GA symptoms, consider SIBO testing when suspicion is high. Treat confirmed SIBO and discuss a gut healthy diet. Beyond AD or psoriasis, there are several other skin lesions or dermatosis suggest a gut link, like hidradenitis suppurativa, cluster with EBD or Crohn's disease, showed microbiome overload. Some small study explore diet, grows yeast, adjunct pre and probiotic, but evidence remains preliminary. And also there is alopecia areata, also like other disease like urticaria or systemic lupus erythematosus, or other like disease from aging or photoaging. Clinical takeaway: prioritize gut barrier and microbiome health. You eat with lots of fiber, consider pre-probiotic as a junk, and coordinate with the gastroenterology when a skin disease happens. So we discuss on the therapeutic side approach to the gut-skin axis. Probiotic, life beneficial microbes, our most practical way to modulate the gut-skin axis. Meta-analysis shows strain-specific modest benefits in adult atopic dermatitis to reduce the severity and psoriasis, with early signal in acne. Mechanistically, selected strain enhance gut barrier integrity compared with pathobionts generate HCFA, such as example including L. salivarius, Acidophilus, rhamnosus, or Longium. Overall, they are generally safe. Expect only mild transient GI effects such as microbiome used as a junk, not replacement for standard dermatology care. The second one is symbiotics. It's a combination of probiotic and probiotics. Fibers that feed the beneficial microbes are gaining interest as well. The rationale is that prebiotic like inulin or fructooligosaccharides can help the introduce probiotic strains established and can also nourish the host resident good bacteria. There was an acne randomized control trial discussed use symbiotic formula and saw not only clinical improvement but also increases in fecal HCFL, indicating the prebiotic component likely to contribute to metabolic benefits. So how about prebiotic and diet in the four skin gut axis? As mentioned, prebiotic are fermentable fibers or nutrients that promote a healthy gut microbiome rather than ingesting live bacteria. One ingest substances that selectively stimulate beneficial bacteria already in the gut. In the context of skin health, a high fiber diet and diets rich in plant-based diversity as a prebiotics. These diets increase production as SCVS like butyrate, which has systemic anti-inflammation effect and has been known to improve epithelial barrier of the skin. So clinically, some dietary modification have shown benefit in inflammatory skin diseases like low glycemic load diets, dairy-free or low dairy diets, high fiber like Mediterranean style diets, or other fermented food like yogurt, kefir, kimchi, and other healthy foods, natural sources of probiotics. It's crucial to note that extremely elimination diet are not recommended unless there is a confirmed food allergy. For patients, they advise you to feed your beneficial bugs. This means consuming prebiotic fibers, possibly resistant starch. That get bacteria metabolized into anti-inflammatory compound. How about fecal microbiota transplantation? Is transfer of stool from a healthy donor to patient gastrointestinal tract with the aim of restoring balanced microbiome. It is an established treatment for recurrent clostridius. So the for psoriasis, no published RCT yet. But case report have described psoriatic improvement after FMT given for Clostridium difference infection. There is also ongoing interest in anecdotally reported cases where psoriasis patient undergoing FMT for other reasons, so expecting clearing of their skin lesion. From a precision medicine perspective, not every patient will respond the same way to microbiome intervention. In the future, we might stratify patients by their microbiome profiles and genetic markers to personalize treatment. Another novel approach such as postbiotic, precision postbiotic, probiotics, or gut-targeted small molecules. So, how about the current guidelines now, 2025? Major dermatology and allergy guidelines are conservative regarding microbiome-based intervention for skin disease. They acknowledge growing research, but generally do not make strong recommendations for probiotic diets or FMT as standard therapy. For example, in 2022, the Academic of Dermatology Association Atopic Dermatitis Guidelines emphasized traditional treatment and note that while probiotics have been studied, results are mixed and no firm recommendation can be made for their routine as in AD. Maybe, hopefully, for the next 5 to 10 years, it will be the future research for the guidelines. How about the research gap? Despite promising findings, many questions remain unanswered, presenting opportunities like how to understand the mechanism of these microbes, how to identification of critical microbes, how to optimize intervention and make it as a protocol for dermatologic disease, and long-term outcome and safety, of course, as a doctor. Personalized approach and biomarkers also a good research gap. And the last, what we have studied is the microbiome of its skin itself. We did a research on the effectiveness of cosmetic product efficacy and its influence on the diversity of facial skin microbiota. Never been done before. It is the first skin microbiome study in Indonesia to effectively result of the formulation. We make a formulation of cleanser and moisturizer with 28 subjects for 1 month. This is the inclusion and exclusion criteria we did at the COVID pandemic in 2021. This is the questionnaire we made to categorize the skin of the subjects. We did the measurement using Korej-Kazaka skin parameters. This is the result of photometer, the elasticity, increase the elasticity of the skin after 28 days, and also reduce of the oily gas, reduce the sebometer production, and also the brightness increase, more glowing, see, corneometer as well, so the dryness is disappeared, thermometer to measure the transepidermal water loss from the skin, also give good result, and also the pH meter. We don't want to make a irritable product, of course, So this is another result. The mexameter for the color. We avoid to be erythema, to increase the redness, to give side effect, This is the result, This is how to collect the skin sample using swab. We test in the labs the extraction, doing the bioinformatic analysis, bacteria metagenomic result, the alpha diversity, microbial diversity index discussion and conclusion. Variables, this is the battery, and we get the heat map film, and before after treatment of 1 month with our the product test of cleanser and moisturizer, there is a difference of bacteria on the subject skin. So there was interesting result, the decrease of bacteroides, Empedobacteria, Peridobacter, Carnivores, Aridobacter, and increase of Pedobacter. This is again the first result of the Indonesian type of skin. So, Alhamdulillah, the product gave a very good result of the clinically, increase the elasticity, good for dry skin, but don't give acne, ya, jerawat, tidak menimbulkan jerawat, increase the brightness without making erythema or redness or side effect, yeah, or other pigmentation side effect. Of course, we don't want to make a side effect like that, yeah. Increase the transepidermal water loss is good for dry skin and tidak mengganggu the pH is nice, yeah. So the bacteria, we found that after 1 month, decrease in Bacteroides, Pedobacter, Peridobacteria, Carnivores, Aridobacter species, while increase the genus of Pedobacter species without involve or getting side effects. Yeah. So, alhamdulillah, we published the paper in World Congress of Dermatology. This is our team, Dr. Caroline. She is also a dermatologist or skin doctor in 2023 in Singapore. And the product already in the market. Yeah, hopefully you can try. It's by PT Paragon or Wardah Cosmetic brand, sister brand. Yeah. So we welcome you to collaborate. Yeah, we propose a collaborative research project. Yeah, to tackle this gap. One idea is maybe like metagenomic or metabolomic analysis of a patient with a particular condition, say psoriasis or something like atopic dermatitis or even acne. Yeah, before and after a microbiome intervention to identify what changes correspond with clinical improvement. Yeah, we can see we have this funding scheme in BRIN. So please feel free to collaborate with us with this ideas. Yeah, of course. Yeah, hopefully it will benefit to our people, our patient, children. Yeah. Our center can conduct controlled preclinical trial, for instance, transplanting human patient microbiomes into germ-free mice to see if develop skin inflammation. Yeah, or we will also interested on running pilot clinical trials. Yeah, with you. So please feel free. We also this is a very good collaboration. Hopefully, probiotic, maybe biologic psoriasis. Yeah. And also we welcome industry partners. Yeah, we have here some. Thank you very much for attending here and doing such as wonderful trials and providing high quality investigational probiotic product maybe and hoping develop new therapeutic formulation is still a blue ocean and a still good opportunity for you. Yeah, this kind of academic industry partnership is essential to move microbiome therapeutic for branch to bedside, regenerative and precision medicine. So in conclusion, the gut microbiome is a frontier dermatology offering exciting possibilities to improve skin health through inside-out strategies. The level of evidence has been steadily rising from anecdotes to controlled trial and even meta-analysis, indicating real benefits in diseases like atopic dermatitis, acne, psoriasis. Yeah. As always, we must integrate innovation with caution, applying only well-supported intervention and doing no harm to the patient. But the trajectory is clear. We can do collaboration in research and innovation in the gut-skin axis will likely yield new adjunct therapies and refine clinical guidelines in the years to come, ultimately leading to more holistic and effective care for patients. By continuing rigorous research, especially collaborative efforts across discipline, we will close the knowledge gaps and unlock microbiome-based solution with the strongest evidence base. This is a compelling example of regenerative and precision medicine action, targeting the body's microbial ecosystem to restore health and achieve more personalized treatment. So thank you very much. Welcome. Contact me anytime. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Reza, for the excellent presentation, very comprehensive. We already heard two fascinating talks about the intricate connection between gut, immune response, and skin health. Okay, I think it's time to begin the Q&A session. I'll read some of the questions. There are two questions. The first one from Dr. Ika. This question goes to Dr. Astri. How does H. pylori manipulate innate signals and antigen presentation to induce tolerance? And can laboratories reliably measure this immune evasion, especially to assess who needs endoscopy? Interesting. Please, Dr. Astri, it's time to respond.
Okay. Thank you for the questions, from Dr. Ika. So actually, the questions is more likely into the bacterial virulence, if how they manipulate the innate signals and the antigen presentations. However, when it's firstly the dendritic cells catch the part of the bacterial antigen of bacterial, and then as definitely our body, the innate immune cells will telling the adaptive immune cells that this is the culprit. We need to finish it, and then we need the specific soldiers coming into that environment. So that's why it will present such cytokine. And then how the pylori, probably the bacterial can also manipulate the epigenetic in the gastric epithelial cells, and then the gastric epithelial cells can produce pro-inflammatory conditions that later affecting into the T cells productions. Firstly, they increase the pro-inflammatory. However, with the higher inflammatory, which means higher PMN, neutrophil, and then it will give a distraction to the epithelial. So what the pylori, and while the pylori itself has urease and then so on, that it can clear the gastric pH. So that's why they increase the inflammatory to give a distraction to the gastric epithelial if it still continue. However, in the first phase, some of the T cells and then also the macrophage can phagocytes and then increase the IL-17 also for the clearance. So that's why if the acute inflammation continues and then the PMN, a lot of neutrophil coming and then distract this what's the epithelial cells deleted and then destruction, and then later it's easily for the replications of pylori and then give a chronic inflammation. And then for the biomarkers, so IL-8 is already well known as the biomarker for ulcer. However, for the IL-17 and then what else that I mentioned before? Interferon gamma, it's most likely into the submucosal. So, sorry, can Dr. Pramita, can you move back into the with the endoscopy?
The question, yeah.
Can reliable measure this immune evasion, especially to assess who needs endoscopy? So it's kind of difficult because who needs endoscopy is more likely to find the bacteria, what the bacteria has done. So if IL-8, because it's similar to IL-6, it's kind of like if there's a lot of inflammation anywhere, so it's definitely high. So it's kind of difficult. But for the IL-17, TNF alpha also coming from the submucosal. So it needs to be taken biopsy. So that's why if assess who needs the endoscopy through the biomarker in the blood periphery, it's going to be difficult. However, probably now people, many studies about the epigenetic, especially for microRNA, because microRNA can be freely circulate in the blood periphery. However, because it's tissue specific, so a lot of different microRNA can be checked. And then some of the microRNA already, but I am not remember clearly. MIR34, and then LAT7, LAT7, it's already well known also for the gastric inflammation.
Thank you, Dr. Astri, for the response. Maybe we move to the next question from Dr. Armedi. This is also for question for Dr. Astri. Based on your experience and research, what is the most important host immune response in gastroenterological diseases? Is there a specific one that is present in every gastroenterological disease, or is it specific only to each type of GI disease? IL-17 and IL-23 are mentioned as being associated with cancer risk. Can this be developed into biomarkers in the future, or are other immune system components still needed? Okay. Would you like to respond, Dr. Astri?
Sure.
Please keep it brief. We only have 10 minutes left. Thank you.
Specific one that is present in every. So because immune response is being everywhere in our body, so and then the cytokine itself usually is pleiotropic, or means it's as dual side. So it means it's the balance that we need to find. So that's why if it's specific, not really, because mostly if you read the studies, it's still controversial that is it really or not. However, for the GI tract, yes, the IL-17, and then also for autoimmune, like what we've done in my lab previously, it's about the encephalitis, and then also IBD, that's the IL-17 that increase. However, we didn't check like, is it IL-17A, 17F, most likely with just IL-17. So because of the pleiotropic, so we cannot really specify. And then so also for the immunotherapy, although we can give some therapy for the inhibitor, cytokine inhibitor, but we must understand that it will give a different effect to different organs of the body, and then probably has a side effect from that. And then IL-17 and then IL-23 mentioned as the, yeah, probably it can be developed into biomarkers as the polymorphism already said that it's correlate and then or not. But we need to check in the phase. I mean, the oncogenic was from the ulcer until cancer. Well, I haven't been studied yet, and I mean, I've never been done to check from the patients, just ulcer until cancer. How about the IL-23? Is it increasing or not? So is it biomarker or not? And then also for if it's cancer, it's already known, but however, in the early stage, we don't know yet. I mean, I haven't doing that research in that early phase of cancer. Okay. I guess that, yeah. And then from Dr. Faisal.
Yeah, Dr. Faisal. The three question goes to Dr. Asri, all of them. In your opinion, what are the main advantages of targeted immunotherapy compared to T-cell-based therapies, especially those derived from autologous stem cell?
Okay, so I already doing my research in that checkpoint inhibitor and then also cytokine inhibitor, but I haven't done about the CAR T, although I've asked for someone who's already in that field, and then they said like it's easier to create the CAR T cells to manipulate it, rather than to choose which cytokines. However, based on the experience that it's easier to get the reagent for inhibitor rather than CAR T, because CAR T we need to taking it out from the patients and then selecting it and then create the specific antigen receptor and then return it back, and then it has side effect. As I know, it's about the neurotoxicity and then so on. However, for the usual, not the usual, I mean, the known now, the immunotherapy, is it the checkpoint inhibitor or cytokine inhibitor? The side effects, it's still manageable, because we already know it's about the balance itself. It's not introducing something new into our body. So that's the main advantage. Is first, it's easier to get, it's cheaper, and then the second one, although it has the side effect, but it's still manageable.
What's manageable? Okay, thank you, Dr. Astri. I think this is the end of the second session. Thank you very much for both of our speakers. Yeah. Dr. Astri Dewayani and Dr. Reza Yuridian for sharing such valuable knowledge and engaging discussion today. It's my honor to present the certificates of appreciation. Huh? Oh, Dr. Reza. Oh, I'm sorry. We already reached the 15-minute discussion. Maybe we can extend the time. This last question from Dr. Already or Dr. Reza. This question was for Dr. Reza. There are 4 questions. Okay, interesting. Given the gut-skin connection, can it be said that people with digestive disorder will definitely experience skin disorders? And the second one, of the human response involved, is there anything that specifically indicates that skin connection? And the third one, does an unhealthy diet or the type of food you eat affect the gut-skin connection? And the last one, if skin disorders such as psoriasis and acne occur, should therapy target the gut or the skin first? Which one is the first one? Okay, please, would you mind to respond this question, Dr. Reza?
Yes, thank you very much, Pak Dr. Ramedi. A very, very good question. So I hope I can answer the question. So the first question is given the gut-skin connection, can it be said that people with digestive disorder will definitely experience skin disorder? So the answer, yeah, is as the research said, there is a relation, but is it definitely? It's not always like that. So gut disorder increase the risk, but are not deterministic. So dysbiosis, permeability, systematic inflammation, and neuroendocrine stress can lower the threshold for dermatosis. But penetration depends on the host factor. Because there are lots of other factors like the genetics, the barrier integrity, the hormonal, the environment. So other factors beside the microbiomes. Hopefully it can answer the first question. So the second question, or the immune response involved, is there anything that specifically indicate gut skin link? There is no single pathognomonic marker yet, but a pattern supports the connection. Like a suite of T cell axis, I have presented of the T helper 2, or other like interleukin 4, interleukin 13 in atopic dermatitis. And in psoriasis, there was a increase of T helper 17 and T helper 1. And decrease of Tregs. So this is quite interesting. Maybe we can discuss a further research topic with the data of that data. So another interesting is the barrier signals endotoxemia is the increase of intestinal permeability such as zonulin or circulating like LPS, like, decreasing the fecal SCFAs. So other can be also a marker for this. Is hopefully I can have answer the second question. The third question, does an unhealthy diet or the type of food you eat affects the gut-skin connection? Of course, what we eat is what the skin, what you appear on the skin. So high glycemic load and some dairy patterns, raise insulin and perturb microbiota. More inflammation and sebum keratinization, like in jerawat, acne, will worsen by oily, spicy food, low fiber food, and processed food, so reducing the the microbes product, or SCFA we discussed later on the presentation, and impair barrier function, relevant, So we have to increase, like more fruits, more vegetable, because they increase, they have a high protein and a high fibers, and good for our guts microbiota. Hopefully it already answered the third question. The last question is, skin disorders such as psoriasis and acne occlusion, should therapy target the gut? Or because it's not yet become the guidelines, although the theory have been 19 years old, 90 years old, but we have to do more research, more RCT, for becoming a guidelines. Like American Academy of Dermatology had not decided that we targeted on giving probiotics as the main or the gold standard of this skin and gut disease link. So, but can be adjunct therapy. Terapi tambahan. So hopefully this can answer all the questions. So thank you very much for a very wonderful question, Pak Dr. Aryan. Thank you.
Thank you for the wonderful response, Dr. Reza. And I think it's the end of the second session. And now it's time for the certificate of appreciation presentation. On behalf of the organizing committee, I would like to express our sincere gratitude to Dr. Astri Dewayani, PhD, and Dr. Reza Yuridian Purwoko, dermatologist. These certificates are a token of our appreciation for your expertise and contribution to this webinar. Once again, thank you, Dr. Astri and Dr. Reza, for your inspiring talks. Okay, finally, I would like to thank all participants for joining this session and for your active engagement. We hope today's discussion provided new perspective. And please stay tuned for our next session. I would like to return the floor to our MC to continue with the next agenda. Have a wonderful day ahead.
Thank you, Dr. Pramita, for leading the session. And I would like also to remind all participants, please fill the attendance form provided by the link in the chat box to get the certificate. Ladies and gentlemen, we have now reached the closing session of today's webinar. To close today's webinar, we would like to invite Head of the Research Center for Preclinical and Clinical Medicine BRIN, Dr. Harimat Hendarwan, to deliver the closing remarks.
Okay, thank you very much, Ms. Andalusi. As this second session comes to an end, I would like to close our webinar today by expressing my appreciation for your presence, attention, and also your participation. Once again, I would like to apologize for the various shortcomings in organizing this meeting, and I hope you enjoyed this webinar and see you next time. Thank you very much and good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam. Thank you.
Waalaikumsalam.
Ladies and gentlemen, that concludes today's webinar. On behalf of the organizing committee, we extend our sincere appreciation to all speakers, moderators, and all participants for their valuable time and active contributions. We hope today's discussion will strengthen collaboration and inspire further research toward precision diagnosis and therapy in gastrointestinal health. Thank you for joining us. Stay healthy, stay inspired, and see you at our next webinar. Have a wonderful day, everyone. Thank you.